Well, good afternoon, everyone. It is a real pleasure for me to be here presenting to you the Pan American Evolutionary Society for Developmental Biology Early Career Award that this year is going to Dr. Deirdre Lyons. So this prestigious award is given to an early career researcher who demonstrates exceptional contributions to the field of Evo-Devo and who has demonstrated the ability to develop an independent research program. And I will show you in a second that Didi fits 100% this award. If my slides move. Okay. So Didi did her bachelor's with Dr. Stan Rakuten in the articulata hypothesis in annelids and arthropods. And then she did her PhD in the Department of Molecular and Cell Biology at the University of California in Berkeley. And she worked with these really cute leeches, which I Googled last night because I'm a plant developmental biologist. But they're really, really cute. And then later, she did her postdoctoral research with Dr. David McClay, and she was working in the gene regulatory networks controlling cellular behaviors in echinoderms. And now she's an assistant professor at the University of California in San Diego, and she's working with everything, all of it together. And basically, she is building up a program in comparative gene regulatory networks in marine invertebrates. So I also want to say that Didi has served her community in many, many ways. She's very generous with her time and resources. She has participated on several committees aimed at increasing diversity, equity, and in general, just inclusion. But she has also served as a research mentor to high school students, to a number of interns. And on top of everything, she currently serves as a mentor to 4 postdoctoral associates, 3 graduate students, 4 undergraduate research assistants, and 2 technicians. So she's basically a superhero, really. So I just want to leave you with a quote that I found on the letter, her nomination letter. And it says, ultimately, what is most striking about Dr. Lyons' work is that it spans systems and questions more broadly than is typical of most developmental biologists. Her ability to take on new systems demonstrates a certain fearlessness, as well as a deep love of developmental biologies. So we should all be more like Didi. So let's welcome Didi.
Thanking the organizers of the meeting and the nominating committee for this incredible honor. I love both of these societies so much. I've been going to them my whole career. I met Dave McClay at an SDB meeting and it influenced my postdoc, and the first Pan Am meeting was the first time I ever had a conversation with a program officer on my own, and really helped me start feeling like a grown-up in science and getting funded. So it's an incredible honor and really wonderful to be able to share some of my research with all of you today. So thanks, and the last thing between you and coffee, I know, so we will try and keep it going. So like many of us, one of the things I'm most interested in Evo-Devo is how you can have 2 embryos that seemingly, when they're young, look similar to one another, like these 2 embryos here that were taken as movies by one of my postdocs, Vanessa Barone, who's here. And from looking at these movies, you might think that these embryos look similar to one another. There's asymmetric and symmetric cell divisions. They have a certain organization with small cells and big cells. But of course, if you are used to working with these embryos, that you know that they are going to be quite different when they grow up. So the embryo on the right is going to turn into an echinoderm. In this case, the sea urchin Lidocynus pictus, and it has characteristics of the echinoderm phylum, like 5-fold symmetry, a water vascular system, and an endoskeleton. It's made of calcium carbonate. And that other embryo that I showed is going to turn into a completely different phylum, the mollusk, in this case, Crepidula atrosolia. And that is going to have bilateral symmetry, overt chirality, an exoskeleton, and other features like a muscular foot. So like many developmental biologists and Evo-Devoists, I've been fascinated by this question for a long time. Starting as an undergrad, how is it that organisms integrate information in their genomes, cell contact and signaling, and signals from the environment to make completely different body plans? And I remain to be really fascinated by that question. So today I'm going to just talk a little bit about those 2 embryos and just make a plug and put in the context that we are, as the very nice, generous introduction mentioned, we are also trying to compare individual species at different distances, at class levels and genus levels, and then in some cases between phyla. And like I don't need to say to this room as well, this involves a lot of tool building and community building and system building. I'm not going to really talk about that explicitly today, but it takes a lot of work to do that, and I know many of you are also doing that. And I hope that some of these species I'm talking about today will be platforms for comparisons that you might be interested in, or new questions that no one has gotten to before. And I just want to make a plug that although you might not be as familiar with echinoderm or mollusk biology or development, that the advent of being able to have CRISPR and better sequencing technologies, and now we're moving towards transgenesis, it's really opening up some exciting experiments. So if in the future you're interested in kind of going into any of these animals, come talk to me and I'll help you connect to an embryo that matches your question. All right, so today I'm going to tell two stories by two people in my lab, Vanessa, who's here, and Grant as well. And Grant and Vanessa were the first people who were brave enough to join a brand new lab. And my first postdoc is Vanessa, and my first graduate student is Grant, and I'm so grateful for their work. And if you would like to hear more about these projects, you can talk to them today, and they will be happy to tell you more about their stories. Okay, and I'd also like to say these two stories are a good example. One, as Dr. Pick said, is curiosity-driven. We weren't looking for it. And the other one is very hypothesis-driven, and we were hoping to go down a very logical route. So we'll see two versions of that. So I'm going to start out with Vanessa's story and with the echinoderms. So just to orient you, we are here in the phylogenetic tree. Echinoderms are a sister group to the hemichordates and within the deuterostomes. And they have 5 extant classes: crinoids, sea stars, brittle stars, sea cucumbers, and sea urchins. And I'm going to start out by talking about the sea urchin body plan and early development. And so while I mentioned that the adults have 5-fold symmetry, pentaradial symmetry, the embryos are bilaterally symmetric. So it's another good example of 2 body plans out of the same genome kind of thing. So sea urchins have been a classic model for understanding development for many reasons, and so I'm just going to very briefly go over some of the important features of the early embryo to understand for the story. The first 2 cell divisions will divide the embryo into 4 equal blastomeres, and these are some of the classical experiments. You can see these colors on the top kind of showing fate maps, a little bit vaguely, determinants between the animal and vegetal pole, and these were some of the first embryological experiments that were done many hundreds of years ago, pulling these blastomeres apart and showing that the information in each one of these blastomeres can actually lead to a perfectly formed normal embryo that grows up. And this is very different than something like what Alice and Edgar talked to you about with ctenophores. You can separate early blastomeres and you do not get any regulation. So these are highly regulative embryos. The third cleavage is going to separate that animal and vegetal poles of each of those blastomeres, and now we're starting to see some differences between cell fate. So experiments that would separate the orange and red parts of the embryo from the blue parts of embryo show that they cannot always regulate. And then the key feature of this embryo for this talk is that in the next division, you will get a highly asymmetric division where these red cells at the very bottom are going to arise from a highly asymmetric cell division, and those are called micromeres. And those cells are important for many reasons. One, they are autonomously specified to make an endoskeleton, so they've been used for lots of experiments in vitro to show that these cells can make a biomineral in the absence of any contacts from the other cells. They also form and serve as important signaling centers to the adjacent cells, telling the cells along the animal-vegetal axis what type of cell they should be. And they also are sister cells to the germ cells. So they've been studied for lots of reasons. And I'll just alert you to the fact that in this picture, you notice that there isn't any information in the left, right, or dorsal ventral axis, and that will become important as well. Those decisions are made a little bit later in the embryo. But here is another movie that Vanessa made showing this asymmetric cell division process. So now we're looking right down the barrel of the vegetal pole of the sea urchin, Lidocanis pictus, and you can see that there are these cells in the center that are the micromeres. And because they're very easy to see, because this cell type arises very early in development, people have been able to study not only the cell biology associated with it, but also the molecular control of how it is specified. So need to have a GRN for a developmental biology talk. So the sea urchin model here now is shown kind of in schematic with the red cells being the primary mesenchyme cells. They undergo a very stereotyped epithelial to mesenchymal transition, which has also been studied as a model. It's the first morphogenetic event in this embryo. And then they will undergo a really interesting set of stereotyped migrations, clustering, and self-fusion to one another, and make a syncytium in which the larval endoskeleton is produced from PMC's kind of a mesoderm, the primary mesenchyme cell tissue. And so because of this very accessible cell determination process and a lot of work from Eric Davidson's lab and the McClay lab and many others, including some of the labs here, there's a very detailed and perturbation-based gene regulatory network for this cell type, going from maternal factors like asymmetrically localized beta-catenin to the early transcription factors that set up the differentiation process all the way down to the biomineral proteins that are embedded in the crystal, as well as some modules that are running things like the cell fusion or the cell migration events. So having, like many of us having model systems like Drosophila or mouse or sea urchins, having this level of detail and then allows us to kind of piggyback off of those discoveries and ask whether specific parts of these networks are changed over the course of evolution. And within the echinoderms, the other model that has been quite well developed is the sea star, Ptera miniata. So I'm showing here, or trying to highlight the fact that they have comparable and yet divergent trajectories, and this is what Evo-Devo people like. You have enough things to compare, but you have enough differences that will make it interesting for you to find the changes in the features you care about. In this particular case, it has been used as a model for understanding many things, including the process of biomineralization. So on the left, this sea urchin makes a bilaterally symmetric larval body plan with a crystal inside, so it kind of looks like this easel or an A that is the biomineral, and the sea star bipinaria larva, which looks similar superficially, has absolutely no skeleton in it whatsoever. So somehow over evolution, we think it's a gain of the ability to make this biomineral a gain of that asymmetric cell division and the program for building a biomineral. And so I'm not going to go into details about that today, but of course, I'd be remiss if I didn't mention that people have then done the hard work to try and understand how that gene network architecture is changing over time. And if you're interested in that, there's tons to understand about it. A point of bringing this up now is just that people are very interested in trying to compare many aspects of sea star and sea urchin biology, and the asymmetric cell divisions is just one of them, but we'd also be interested in understanding how the patterning along any axis or the development of different tissues comes about, and there are many labs that are working on those processes. So if that's something you're interested in, there's a ton to read about. But today I'm going to focus on one observation that Vanessa made when she was looking at these two embryos. So Vanessa and I met at the embryology course, and she was shopping for a set of embryos where she could study cell contact dependent signaling in an evolutionary context. So she was interested in using sea stars and sea urchins. So here, these are again 2 movies that she made. On the left is a sea urchin embryo. Now the asymmetric cell division is on the bottom, so those micromeres are at the bottom. And you can see that fourth cell division in the embryo is highly asymmetric, giving rise to these micromeres and their sister cells called mesomeres, which is the second to bottom layer of cells. In the sea star on the right, Vanessa was interested in the fact that these embryos are the same cell stage, so these are both 16 cell stage embryos. They're the same time in development. The sea star has much less contact between the cells. And so while Vanessa came to study that process, and she gave a great poster the other day about how beta-catenin and cell signaling might be contributing to regulating cell fate, she also noticed the fact that even though sea stars are famous for having symmetric cell division and not having this asymmetric cell division, then in fact there were some cases, let's see if I can get my pointer to work this time. There were some cases where there would be a cell that is smaller than another cell, and in some cases the asymmetry between 2 embryos at the 16 cell stage can almost be twice as much. Some embryos are twice as large as others, so this kind of went against the idea that there was only symmetric cleavages in the sea star. So this is what Vanessa noticed this, and then she applied what someone I met when I was at Friday Harbor as an undergraduate, George Van Dasso, called LCPA analysis, which you should always be doing, which is to look closely and pay attention. And so Vanessa decided, instead of ignoring that, to go into that deeper and ask, what could that be? Why is that does that have any significance? So the way Vanessa approached this was to label those cells that were smaller by doing a photoconversion experiment. So she injected mRNA coding for the photoconvertible protein CATA into the egg, let them grow up into a 16-cell stage about, empirically determined which was the smallest cell by confocaling the embryo and deciding and measuring and doing a quick measurement of which one was a smaller cell, and then photoconverting that cell. And then she wanted to know, what do these small cell clones end up making in the embryo? Is it random, or there's some type of pattern? So by using this technique and then growing the embryos up to the first time that you can really determine the dorsal ventral left, right, anterior, posterior axes carefully, which is at this 72-hour time point, Vanessa found that the clone often would be in the ventral anterior portion of the embryo. So she did this once and then kind of made these cartoons, which we'll be looking at in the next slide, that summarize where that pattern was that she saw. And then she did it many times and found that there is a bias, that the small cell clone is biased towards the ventral half of the anterior ectoderm. And as a control, to show that this is different than just random injection, she went about just randomly injecting blastomeres, doing this type of the photoconversion, growing them up, scoring them, and found that a randomly injected blastomere is statistically different than the ones where she focused on the smaller cells. So this was kind of the first evidence that there might be some type of asymmetry, both along the animal vegetal and dorsal ventral axis in a sea star. And even though in the literature some people had mentioned that there were asymmetric cell divisions, no one had really pursued it with this level of analysis. So that's what Vanessa did. And then from that, then she had a couple other questions that were related to that. So for instance, does the smallest cell influence dorsal ventral patterning? And then Vanessa went about these technically challenging experiments that kind of mimic the classical experiments that have been done in sea urchin, where she could separate the cells of the 2-cell stage, the 4-cell stage, the 8-cell stage, and on, and then just looked for what those embryos are going to make. And of course, the paradigm from sea urchins is that you should be able to separate these early blastomeres up to the 4-cell stage, and they should make perfectly formed but smaller sea urchins. And so that's exactly what Vanessa found, that she could, as a control, relative to the controls on the left that are the normal sized embryo, if you separate the cells at the 2-cell stage or at the 4-cell stage, you make generally perfectly normal embryos, and that is shown here on this graph. It's just the first 3 lines where you see most of them doing perfectly fine, being separated at the 4-cell stage. But then when you start separating the animal and vegetal halves, you start seeing defects. So the animal half will make kind of an ectodermal embryo with it's ciliated, but it can't make a gut or endoderm. And the vegetal cells are often able to regulate, and this is exactly what happens in the sea urchins. So from that experiment, she showed that we're mimicking some of the behavior of sea urchins, but that it didn't seem as though that cell that's in the animal pole that might be smaller would be inheriting any special information for making the dorsal ventral axis, because you would expect that if that cell in the animal pole were inheriting some type of maternal factor, that in the animal separated organisms experiments that you might be able to see some type of normal development of the gut or the dorsal ventral axis, but she did not see that. So we don't think that it's a maternally localized difference that's influencing the asymmetric cell division or imparting this entrainment to dorsal ventral axis. And then Vanessa went on to do a number of other very technically challenging and elegant experiments, and I'm just going to show you one of them. But then she said, well, maybe it's not that it's something maternally inherited, maybe it's something that's different about that cell by the virtue of it being the smaller cell, that after the maternal factors are gone, some type of zygotic cascade that happens that makes that cell different, that entrains the dorsal ventral axis. So then she did another technically challenging set of experiments. So in the controls that are shown on the top, she just injected one of the blastomeres with the lineage tracer and grew it up to the time when you can score dorsal ventral left and right, anterior posterior, and showed that a randomly injected embryo that's not manipulated doesn't have any bias in its pattern of where it's going to show up. And then she experimentally reduced a set of those embryos by making them smaller and asked, if I experimentally make that cell smaller, will it influence the dorsal ventral axis? And she found that there actually isn't any statistically different relationship between that experimentally smaller cell and the dorsal ventral axis, which is shown here in this bottom rows plot. And even though it might look like some of the blue is biased to the ventral side, it wasn't statistically different. So it doesn't seem like making the cell smaller on its own also is in training the dorsal ventral axis or influencing it. So what she found from these experiments and others that are explained in her paper, she found that these small cells, although they're predictably presaging or predicting where the dorsal ventral axis is going to be, and they're the first morphological indication of dorsal ventral axis that we can see in any echinoderm that's looked at so far, that neither maternal determinants or segregation of the small cells seem to, or the fact that the cells are small, seem to be required to induce the embryonic axes. So the mechanism that's relating these cell size asymmetries to the patterning at a molecular level and experimental embryology level still remains at large. But what's exciting about it for us is that the timing of this process is comparable to sea urchins, and although we cannot tell the dorsal ventral axis from looking at the sea urchin embryo, there have been a few cases where molecules that are expressed on the dorsal side of the embryo are coming up by in situ hybridization up around the same time that this decision is being made. So it's possible that Vanessa's kind of stumbled upon a conserved process of how the dorsal ventral axis is being determined based on something that we have yet to figure out. And there have been a lot of hypotheses about what those things could be, like redox reactions or sperm entry point or other types of things, but the field remains with a good solid hypothesis about how that's working. There are many people who are looking at the gene regulatory networks that are integrating the anterior-posterior pattern in dorsal ventral. Things, for instance, Ryan Range's lab, who's also here at the meeting, so if you're interested in talking to us more about that, you can. And then a bonus that Vanessa got out of these experiments was that she found that the fate map of sea stars and sea urchins are actually different. And in the field, people have just assumed that the embryos look so similar to one another that a lot of the fate mapping experiments are the same. So she found out that they're not, and we're excited to think about that in terms of some of the differences in fate determination that we know about in sea stars and sea urchins. So if you'd like to talk to Vanessa more about that, she's here and would love to discuss that with you. And now in the remaining time I have, I'm going to give a little bit about this other project by Grant. And unlike Vanessa's project, this was, that was Vanessa's side project while she was doing the experiment she was most excited about. But Grant came to my lab very excited to understand the evolution of biomineralization. So a completely different topic. So here's our hypothesis-driven one. And both Grant and I are very fascinated by the process of biomineralization, which happens in a really exciting and diverse way across not only the animal kingdom, but all kingdoms of life. So I'm just showing a couple maybe familiar instances of biominerals that have some value to humans and sometimes get in our way. Like the top right picture is a hull of a ship that's covered by annelids that are making a tube that is going to hold down the, obviously prevent the speed of the boat. And so there's practical reasons where we want to get rid of biominerals sometimes. There's other reasons where they're quite precious to us and useful, like pearls. And there's also kind of impending worries about how fisheries around the world are going to be able to survive with ocean warming and pH changes as the oceans are warming, as biomineralization really matters, really takes, sorry, I'm nervous. The process of biomineralization really requires differences in pH between the inside of the cells and the outside of the cells. And so if you're increasing heat and decreasing pH, you're going to have some real problems with making biominerals. And as a lot of fisheries are food sources for the world, there are some real threats about that and interest in trying to control that process and get ahead of it. People are also very interested in using biominerals as biomimetic tools. So there's cool bacteria that make magnetic biominerals. And so there's a lot of interest in biomaterials science. And of course, there's lots of interest for us, both for conservation and for health, to understand how biominerals form. Okay, so here's just a couple of examples, and not only are there these practical reasons to be interested in biomaterials, there's just the curiosity-driven, cool Evo-Devo questions about biomineralization. So here's the one that I find most interesting. So if you look at the fossil record, and if you look at, incorporate data from clocks about, evolutionary clocks about how long ago animals diverged, and if you look at the distribution of different types of biominerals across the animal tree, here this phylogeny is showing animals that have silica-based biominerals, calcium carbonate biominerals, calcium phosphate biominerals, and you can see that these different types of biominerals are distributed along the tree, interspersed with not only groups that don't make biominerals, but biominerals of a different type. And so from just the phylogeny work, it seems as though there isn't a one invention of particular types of biominerals, but it happened many times. independently. And if you look at the clock, it also seems as though this biomineralization process happened right at that Ediacaran to the Cambrian transition. So when people refer to the Cambrian explosion, sometimes they talk about it as life getting hard, because it's not necessarily only the biology was changing so much, the biomineralization somehow synchronously was happening in each of these lineages, and so we have a great fossil record for it. And I'm happy to debate ideas about what that might mean and how we got that way. But it's a really nice place to look at the convergent evolution of a structure across many, many lineages. And that's kind of one of the reasons that I'm interested in it. I'm also interested in it because mollusk shells are characteristically beautiful and complex, and I'm very interested in how you might modify this biomineral process in organisms like mollusks. So mollusks are the second most specious group of animals, and they have iconic shells in these different groups and do really spectacular and interesting things with them. And I think that they're a great Evo-Devo model. Okay, so now with the remaining time I have, I'm just going to go over some of the things we're trying to, like how do we get a handle on how the shell evolves, and using mollusks as an example. So I'll just tell you from many years of people studying mollusk shells and biomineralization in many different contexts that you kind of need a few, I think, these four main things to understand how you make up a biomineralization program. So one thing is that you need to be able to make the biomineral itself, and the biominerals, when we talk about that, is a biologically controlled crystallization of minerals, and many of them are calcium carbonate. And so what's actually happening is that calcium carbonate has been made by organisms long before animals existed, and it's kind of a byproduct of dealing with being in the ocean where much of life evolved. And so the going theory is that some amount of the proteins that are necessary for biomineralization or genes are actually just an exaptation of ancient processes for regulating pH and calcium carbonate. So some percentage of the proteins involved are going to be from this category. And then there's going to be some category of proteins that are going to be what we would call taxon-restricted genes. So when people have done proteomics and transcriptomics on biomineralizing tissues, you get a list of a lot of proteins that have no homologs to any other proteins. And in many cases, there's a really high percentage of genes that have no homolog, even between sister species. So the idea is that this is a place where cells are promoting or somehow harboring a lot of genetic variation, and genes are being evolved very rapidly or diverging very rapidly. And some examples of how that happens would be domain shuffling, for example, and also the characteristic nature of the particular proteins that are involved in biomineralization. So for instance, there are a lot of proteins involved that have intrinsic disorder. So this is just showing a little diagram of a protein that has no structure to it, completely intrinsically disordered in A, and then B is an example where you could mix an intrinsically disordered domain with a structured domain, and then some proteins that have absolutely no intrinsic disorder at all. And intrinsically disordered proteins I think are becoming more and more interesting to study. They're a little harder because we can't predict their function from their topology, and they're harder to crystallize and look at in the traditional ways we think of looking at proteins, but they seem to be involved in some really, really interesting processes, including biomineralization. And then for me, the last thing is that somehow all of these things need to be integrated into our developmental gene regulatory networks that we know about. So for instance, I mentioned that the sea urchin embryo makes an endoskeleton, so that biomineral that's being formed is within a mesodermal cell, and it's actually inside a syncytium, really inside the embryo. While the mollusk shell is made from ectoderm and is put completely on the outside of the animal. So how is it that you would coordinate these first three I talked about and then put them in specific places? So that's kind of what I'm interested in trying to understand. And so if you're interested in doing this in mollusks, here's kind of the starting point. There's about 50,000 living species of mollusks, so they're quite diverse. So which mollusks do you start with? Out of those many, many, only about 45 species, 50, maybe up to 100, depending on how long ago we checked the literature, have any shell proteome or mantle transcriptome. So the mantle is the tissue that's secreting the shell. So the lists of proteins and mRNAs that are expressed in that tissue are only a fraction of the diversity that exists out there. From those lists, though, it's better to have 45 than none. We know about 4,000 to 5,000 shell matrix proteins. That's what we call those proteins that are controlling biomineralization. So many of them have been identified. But we've kind of in this holding pattern where we can keep on sequencing things, right? But to make the next leap to function has been a lot harder. So I'd say out of these 5,000 shell matrix proteins, only about a dozen of them have ever been used in any type of heterologous situation where you're making them in bacteria and letting them interact with minerals and testing whether they actually do organize biomineralization forms. And only about 10 of those have ever been looked at functionally in vivo. So very few kind of developmental biology experiments have been done on the SNPs where you're knocking genes down. And 0 developmental GRNs have been made for biomineralization within mollusks. So I would like to try and get at making some of these statistics a little bit better. And I would argue that I'm picking a good species to start with. This is the slipper snail Crepidula, which is the model system in my lab. Conklin worked on it for his PhD at the MBL, and I'm very privileged to have started to work with it at the MBL, right where he was doing it, to understand these embryos. And they have large shells, they're large animals themselves, so easy to dissect some of those tissues out and get the biomaterial that we need to understand the biominerals, and they make many, many, many embryos, and the nice thing about them is we really understand their development quite well. So here's a couple of Conklin's pictures on the bottom showing stages of shell gland formation, so the beginning of the process of biomineralization. And he showed that it's that this process happens at a particular time that we can find in this embryo, and other experiments have shown that this process is kind of like a gastrulation event. The ectoderm will invaginate and then form a protected space across which the biomaterial starts to be deposited, and then that shell gland will actually evaginate afterwards and get larger and larger. So it's kind of actually a cool comparison to morphogenetic events on the ventral side of the embryo where their mouth forms. And the nice thing about Crepidula, really the most important thing, is that we can inject them and transform them with mRNAs and reagents for CRISPR and plasmids and other things like that. That's the real advantage of this mollusk over others. So here's a movie that I made of shell gland formation at the same time that we knew Conklin had found shell gland formation happening, but in the live embryo. And I wish I could show him all the things we've learned, because I think he'd be happy to know we could piggyback on his excellent observations and see new things. So what we wanted to know is, can we identify shell matrix proteins in an animal where we could potentially knock them down and perturb them and do all those experiments that haven't been able to be done? And can we identify there are any in the embryo? So we looked for the matrix proteins in the adult because the tissue is easier to work with, but could we get access to their biology in the embryo? And we were also interested in their evolutionary history. How old were they? So you don't even need to look at this picture. This is just basically just the title here. We did proteomics on the adult shells, and we did differential expression between the mantle tissue and other tissues, and we found 185 shell matrix proteins in the adult, and 20 of those were differentially expressed in the mantle tissue. So these are the ones that we wanted to focus on. As I mentioned, we think that a lot of biomineralization is exapted from other processes, so we expect that some of the genes that are in the shell matrix are also expressed in other parts of the animal. So this is one of those slides that you can't read all the details, but this is just to show we did the clustering to find out what genes were upregulated in the mantle, and we found a number that were not in SNPs, just ones that are differentially expressed, probably acting intracellularly in the embryo. And I just want to emphasize the fact this is what happens when you work with mollusks oftentimes, is you're looking for your list. Like if you were expecting a GO-term analysis next, it's not coming, because most of the top hits will be something like no blast hit, no blast hit, no blast hit, no characterized protein, uncharacterized protein, so we had to kind of go at it some other way. So what we did was we just looked at the differentially expressed genes and the highest ones, and we picked, for instance, the one that was most highly expressed. And it turns out to be a protein that has a realer domain, has a lot of intrinsic disorder, so that we called it very creatively SNP1, because it's the most highly expressed one. And then it turns out that if we do a different type of analysis, so this had no blast hits when we just did pairwise comparisons, but if you use something like orthofinder analysis, which is doing all-by-all comparisons with a much larger group of organisms, we actually found that this SNP1, which we initially thought was lineage restricted, was only in Crepidula, actually is in other species. And I'm just going to I will go through this quickly by saying if you use this orthofinder analysis, I'm working with another postdoc in the lab, Jessica, who talked about nudibranchs today, we can find the distribution of these differentially expressed SNPs across the tree. And so some of them definitely are Crepidula specific, but some of them are shared back to gastropods, and some of them are shared back to all metazoans. So we're excited to find the the evolutionary trajectory of that. And then we started screening them in the embryo, and at least 5 of these are expressed in the shell gland. So these are kind of our top hits as we go through that list, screening them to find ones that we could now go after with CRISPR and perturb and hopefully start to build a gene regulatory network in a mollusk that someday might be comparable to something like a sea urchin, and to really get at the biology of the SNPs themselves. So with that, I will thank the rest of my lab and my funding sources, and thank you again for the award, and everybody can get coffee now.